Hello, I'm SA, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
Linus Carl Pauling, a name synonymous with scientific innovation and, later, passionate advocacy, first emerged as a prodigious intellect captivated by the fundamental forces governing the material world. His initial fascination resided within the realms of chemistry and physics, a dual attraction that would ultimately coalesce into his groundbreaking investigations into the very nature of the chemical bond. Pauling's early work distinguished itself through the innovative application of quantum mechanics to chemical bonding, a field then struggling to move beyond simplistic electrostatic models. The prevailing understanding of chemical bonds before Pauling often relied on visualizing them as purely ionic attractions, where electrons were fully transferred between atoms, or purely covalent interactions, where electrons were shared equally. These rudimentary models, while useful in some contexts, proved inadequate for explaining the diverse range of observed molecular properties and behaviors. A more sophisticated framework was required to account for the nuances of electron distribution and the energetic considerations that dictated molecular stability. Pauling recognized that quantum mechanics, a revolutionary theory describing the behavior of matter at the atomic and subatomic levels, held the key to unlocking a more complete and accurate depiction of chemical bonding. His approach involved employing the principles of quantum mechanics to approximate solutions to the Schrödinger equation for molecules. This equation, the cornerstone of quantum mechanics, describes the behavior of electrons in atoms and molecules, but its exact solution is mathematically intractable for all but the simplest systems. Pauling, therefore, utilized approximation methods to gain insights into the electronic structure of molecules. This led to the development of his valence bond theory, a conceptual framework that emphasizes the formation of chemical bonds through the overlap of atomic orbitals. A pivotal aspect of Pauling's valence bond theory was the introduction of the concept of orbital hybridization. Atomic orbitals, which describe the spatial distribution of electrons around an atom, possess distinct shapes and energies. Pauling proposed that these atomic orbitals could mix or hybridize to form new hybrid orbitals with different shapes and directional properties better suited for forming strong and stable chemical bonds. For example, he postulated the existence of sp squared, and sp cubed hybrid orbitals, each arising from the mixing of different combinations of s and p atomic orbitals. Consider the specific instance of methane, a simple yet fundamental organic molecule with a formula CH4. Prior to Pauling's work explaining the tetrahedral geometry of methane with its 4 equivalent carbon-hydrogen bonds, presented a challenge. Carbon has 2 s electrons and 2 p electrons in its outer shell. Simple overlap of these orbitals with the s orbitals of hydrogen atoms would predict bond angles of 90 degrees, corresponding to the orientation of the p orbitals. This prediction stood in stark contrast with the experimentally observed bond angles of approximately 100 and 9.5 degrees. Pauling's concept of sp cubed hybridization provided an elegant resolution to this discrepancy. He proposed that the 1s orbital and 3p orbitals of carbon hybridize to form 4 equivalent sp cubed hybrid orbitals. These hybrid orbitals are arranged tetrahedrally around the carbon atom, pointing towards the 4 hydrogen atoms. The overlap of these sp cubed hybrid orbitals with the s orbitals of the hydrogen atoms results in the formation of 4 equivalent and symmetrically arranged carbon-hydrogen bonds, perfectly accounting for the observed tetrahedral geometry of methane. This specific calculation of bond angles in methane using hybridized orbitals stands as a striking early success of Pauling's approach. The intellectual environment of the time significantly shaped Pauling's thinking. Gilbert N. Luce's work on electropairing, particularly his concept of the covalent bond as a shared pair of electrons, served as a crucial foundation. Luce's emphasis on the importance of electron configurations in determining chemical properties resonated deeply with Pauling, inspiring him to seek a quantum mechanical explanation for Luce's empirical observations. Luce's dot structures, while insightful, lacked a rigorous theoretical underpinning, a gap that Pauling sought to fill with his quantum mechanical approach. Further, the burgeoning field of quantum mechanics itself, spearheaded by luminaries such as Werner, Heisenberg, and Erwin Schrödinger, provided the theoretical tools and intellectual impetus for Pauling's investigations. The development of wave mechanics, with its description of electrons as waves rather than classical particles, offered a novel perspective on atomic and molecular structure. Pauling skillfully adapted these quantum mechanical principles to the specific problem of chemical bonding, forging a powerful new approach to understanding molecular properties. The early quantum mechanics community, with its radical ideas and mathematical formalisms, established the intellectual bedrock upon which Pauling constructed his valence bond theory. The initial, often abstract, Formulations of quantum mechanics were gradually being applied to concrete chemical problems, and Pauling was at the forefront of this transformative process. Pauling's early scientific focus was therefore sharply defined to develop a quantum mechanical understanding of the chemical bond that could accurately predict and explain molecular properties. He tackled the existing problem of the inadequate bonding models by applying the newly developed principles of quantum mechanics, particularly the Schrödinger equation, to approximate the electronic structure of molecules. This led to the formulation of valence bond theory and the revolutionary concept of orbital hybridization, exemplified by his successful calculation of bond angles in methane. The influence of Lewis's electron pairing ideas and the broader quantum mechanics community, including figures like Heisenberg and Schrödinger, provided both the conceptual inspiration and the theoretical framework for his groundbreaking work. His early work laid the groundwork for a more nuanced and accurate understanding of chemical bonding, moving beyond simplistic electrostatic models and paving the way for future advancements in the field. Pauling's early explorations into the quantum mechanical nature of the chemical bond revealed the limitations of existing models in fully explaining the diverse properties of molecules. A significant challenge lay in understanding the varying degrees of ionic character exhibited by different chemical bonds. Purely covalent models, where electrons are shared equally, and purely ionic models, where electrons are completely transferred, represented idealized extremes. In reality, most bonds exist somewhere in between, displaying a partial ionic character that influences molecular properties such as dipole moments and reactivity. Pauling recognized the need for quantitative measure of an atom's ability to attract electrons within a chemical bond, allowing for a more accurate description of bond polarity. To address this problem, Pauling developed the concept of electronegativity, a property that quantifies the power of an atom in a molecule to attract electrons to itself. His approach was ingenious, grounded in the meticulous analysis of thermochemical data, specifically bond energies. He observed that the actual bond energy of a heteronuclear diatomic molecule, such as hydrogen chloride, invariably exceeded the average of the bond energies of the corresponding homonuclear diatomic molecules, hydrogen and chlorine. This extra bond energy, he reasoned, arose from the ionic resonance energy associated with the partial ionic character of the bond. Pauling's method involved comparing the bond energy of the hydrogen-hydrogen bond with the bond energy of the chlorine-chlorine bond. He then contrasted this average with the bond energy of the hydrogen-chlorine bond. The hydrogen-hydrogen bond energy is approximately 436 kilojoules per mole. The chlorine-chlorine bond energy is approximately 242 kilojoules per mole. The hydrogen-chlorine bond energy, however, is significantly higher at approximately 431 kilojoules per mole. Pauling attributed this difference to the additional stabilization arising from the partial ionic character of the hydrogen-chlorine bond, where chlorine attracts electrons more strongly than hydrogen. From these observed differences in bond energies, Pauling derived a scale of electronegativity values. He assigned an arbitrary value to one element, initially hydrogen, and then calculated the electronegativities of other elements relative to this standard. The difference in electronegativity between two bonded atoms was directly related to the square root of the ionic resonance energy. A larger electronegativity difference implied a greater degree of ionic character in a more polar bond. This provided a quantitative framework for predicting the direction and magnitude of bond dipoles, and thus the overall polarity of molecules. Irving Langmuir's work on isoelectronic structures exerted a discernible influence on Pauling's thinking. Langmuir's emphasis on the electronic configuration of molecules, particularly his depiction of shared electron pairs completing the octets of atoms, resonated with Pauling's quest to understand electron distribution in chemical bonds. Langmuir's ideas, while predating the full development of quantum mechanics, highlighted the importance of electron arrangements in determining molecular properties, providing a conceptual bridge to Pauling's quantum mechanical treatment of bonding. Earlier still, J.J. Thomson's discovery of the electron and his subsequent proposals regarding its role in chemical bonding provided a foundational cornerstone for all later work in this area. Thomson's model, though rudimentary by modern standards, posited that chemical bonds arose from electrostatic attractions between atoms and electrons. This early insight, emphasizing the electron's central role in chemical interactions, profoundly shaped the trajectory of chemical bonding theory, ultimately leading to Pauling's sophisticated electronegativity concept. Robert Mulliken later refined Pauling's concept of electronegativity by providing an alternative scale based on atomic ionization energy and electron affinity. Mulliken defined electronegativity as the average of the first ionization energy and the electron affinity of an atom. Ionization energy represents the energy required to remove an electron from an atom, while electron affinity represents the energy released when an electron is added to an atom. Mulliken's approach offered a more direct connection between electronegativity and fundamental atomic properties, providing a complementary perspective to Pauling's thermochemical approach. While Mulliken's scale yields numerical values that differ from Pauling's, the underlying principle remains the same. Electronegativity reflects an atom's ability to track electrons in a chemical bond. The Mulliken scale finds utility in computational chemistry and theoretical calculations. Pauling's electronegativity scale quickly became an indispensable tool for chemists. Its utility lay in its simplicity and predictive power. By simply comparing the electronegativity values of two bonded atoms, chemists could readily estimate the polarity of the bond and anticipate its reactivity. A large electronegativity difference suggests a polar bond, making the molecule susceptible to nucleophilic or electrophilic attack. Conversely, a small electronegativity difference suggests a less polar bond, indicating different reactivity patterns. The concept of electronegativity continues to be a cornerstone of modern chemistry. It is a fundamental concept taught in introductory chemistry courses worldwide, providing students with a basic framework for understanding chemical bonding and reactivity. Beyond the classroom, electronegativity finds widespread application in diverse areas of chemistry, including drug design, materials science, and catalysis. For example, in drug design, understanding the electronegativity of different functional groups is crucial for predicting how a drug molecule will interact with its biological target. Similarly, in materials science, electronegativity differences play a vital role in determining the properties of semiconductors and other advanced materials. Electronegativity serves as a powerful predictive tool. Chemists use it to anticipate the polarity of bonds, the direction of dipole moments, and the likelihood of chemical reactions. It is a simple, yet elegant, concept that provides a bridge between the abstract world of quantum mechanics and the tangible reality of chemical behavior. Pauling's introduction of electronegativity marked a major step forward in our comprehension of the chemical bond, providing a quantitative framework for understanding the diverse properties of molecules. It allowed chemists to move beyond simplistic models and embrace a more nuanced and accurate depiction of chemical bonding, one that continues to shape our understanding of the chemical world today. The enduring legacy of electronegativity lies in its continued relevance and widespread use in modern chemistry, confirming its status as one of Pauling's most influential contributions. Pauling's exploration of the chemical bond, particularly his articulation of electronegativity, provided him with a powerful lens through which to view the complexities of biological molecules. He then turned his attention to the intricate world of proteins, tackling a challenge that stood as a major barrier to biological understanding and determining the three-dimensional structures that dictate protein function. The quest to understand protein structure was a formidable undertaking in the mid-20th century. Proteins, large and complex molecules composed of chains of amino acids, perform a vast array of functions within living organisms, from catalyzing biochemical reactions to transporting oxygen. However, their function is intimately linked to their three-dimensional shape. To truly understand how a protein works, scientists needed to know precisely how its polypeptide chain folded into a specific, intricate architecture. Pauling approached this problem with a combination of theoretical insight and hands-on model building. He began with a thorough understanding of the peptide bond, the chemical link that joins amino acids together in a polypeptide chain. He recognized that the peptide bond possessed partial double bond character, a consequence of resonance, which restricted rotation around that bond and constrained the possible conformations of the polypeptide backbone. Building upon this foundation, Pauling employed X-ray diffraction data, a technique that provides information about the arrangement of atoms in a molecule based on how X-rays are scattered with a sample. While X-ray diffraction could provide valuable clues, the interpretation of the data for such complex molecules as proteins was a difficult task. Pauling's genius lay in his ability to combine the X-ray data with his deep understanding of chemical principles and his innovative use of model building to deduce likely structures. Pauling and his collaborators meticulously constructed physical models of polypeptide chains, using precise measurements of bond lengths and angles derived from known crystal structures of small molecules. They then explored the possible conformations of the chain, guided by the constraints imposed by the peptide bond and by the principle that atoms should be able to approach each other without encountering steric clashes. This process of systematic model building, informed by X-ray diffraction data and chemical principles, led Pauling to propose two fundamental structural motifs of the alpha helix and the Beer sheet. The alpha helix is a coiled structure resembling a spiral staircase in which the polypeptide backbone winds tightly around an imaginary axis. The structure is stabilized by hydrogen bonds between the carbonyl oxygen of one amino acid residue and the amide hydrogen of an amino acid residue 4 positions down the chain. The Beer sheet, in contrast, is an extended structure in which polypeptide chains align side by side, forming a sheet-like arrangement. The chains can run in the same direction parallel beta sheet, or in opposite directions antiparallel beta sheet. Hydrogen bonds between the carbonyl oxygens and amide hydrogens of adjacent chains stabilize the beta sheet structure. Pauling's publication of the alpha helix structure in 1951 was a landmark achievement. The structure was immediately recognized as a fundamental building block of many proteins. The alpha helix provided a compelling explanation for the observed X-ray diffraction patterns of certain fibrous proteins, such as keratin, the main component of hair and wool. The development of these models owes a debt to prior work, particularly that of William Asbury. Asbury, a British textile physicist, pioneered the use of X-ray diffraction to study biological fibers, including wool and hair. His X-ray patterns suggested a repeating structure along the fiber axis, which he termed the alpha configuration. While Asbury's interpretation of the alpha configuration was ultimately incorrect, his work provided initial evidence for a regular, repeating structure in proteins and inspired Pauling to pursue a more detailed structural analysis. The initial reception of Pauling's alpha helix model was generally positive, although some skepticism existed. The idea that proteins could adopt such regular, well-defined structures was a novel concept at the time. However, the subsequent work of Max Perutz and John Kendrew, who determined the detailed three-dimensional structures of myoglobin and hemoglobin using X-ray crystallography, provided strong support for Pauling's predictions. Perutz and Kendrew demonstrated that these globular proteins contained significant amounts of alpha helix structure, confirming its importance as a fundamental structural element. The alpha helix and beta sheet are ubiquitous structural motifs found in a vast array of proteins. They form the building blocks of more complex protein architectures and play a crucial role in determining protein function. For example, the oxygen-carrying protein hemoglobin contains multiple alpha helices that create a hydrophobic pocket for the heme group, the molecule that binds oxygen. Similarly, many enzymes utilize beta sheets to form the active site, the region of the protein that binds to the substrate and catalyzes the chemical reaction. These structural motifs are studied extensively using a variety of modern techniques, including X-ray crystallography and cryo-electron microscopy. X-ray crystallography, the technique used by Perutz and Kendrew, involves bombarding a crystallized protein with X-rays and analyzing the diffraction pattern to determine the arrangement of atoms within the protein. Cryo-electron microscopy, a more recent development, allows scientists to visualize proteins at near atomic resolution without the need for crystallization. These techniques have confirmed the widespread prevalence of alpha helices and beta sheets in proteins and have provided detailed insights into their structure and function. The enduring legacy of Pauling's work on protein structure lies in its profound impact on our understanding of biology. The alpha helix and beta sheet remain fundamental concepts in structural biology and are essential for comprehending protein folding, protein function, and the molecular basis of disease. His insights paved the way for the development of modern structural biology and have had a transformative effect on fields ranging from medicine to biotechnology. The structures he proposed are taught in introductory biochemistry courses worldwide, and they serve as the foundation for countless research investigations aimed at understanding the intricacies of life at the molecular level. His work allows scientists to design new drugs, engineer proteins with novel functions, and develop new materials with tailored properties. The impact of the structural insights continues to resonate throughout the scientific community. The enduring legacy of Pauling's work on protein structure lies in its profound impact on our understanding of biology. The alpha helix and beta sheet remain fundamental concepts in structural biology and are essential for comprehending protein folding, protein function, and the molecular basis of disease. His insights paved the way for the development of modern structural biology and have had a transformative effect on fields ranging from medicine to biotechnology. The structures he proposed are taught in introductory biochemistry courses worldwide, and they serve as the foundation for countless research investigations aimed at understanding the intricacies of life at the molecular level. His work allows scientists to design new drugs, engineer proteins with novel functions, and develop new materials with tailored properties. The impact of his structural insights continues to resonate throughout the scientific community. Pauling's intellectual curiosity extended beyond the realm of protein structure, leading him to apply his understanding of molecular architecture to the study of disease. He sought to understand the molecular origins of illness, particularly those with a genetic component. This pursuit culminated in groundbreaking work on sickle cell anemia, a disease that provided a crucial link between genetics and molecular biology. The problem Pauling addressed was the understanding of the molecular basis of genetic diseases. Prior to Pauling's work, genetic diseases were understood primarily as inherited conditions with unknown underlying mechanisms. While the role of genes in transmitting these diseases was appreciated, the connection between genes and specific molecular alterations remained elusive. Physicians observed patterns of inheritance, yet they lacked the tools to pinpoint the precise molecular defects responsible for these conditions. Pauling recognized that understanding the molecular differences between healthy and diseased individuals could unlock new avenues for diagnosis and treatment. Pauling's methodology involved demonstrating that sickle cell anemia is caused by a specific alteration in the hemoglobin molecule. He hypothesized that the genetic mutation could lead to a change in the structure of a protein, which in turn could disrupt its function and cause disease. To test this hypothesis, Pauling and his colleagues employed electrophoresis, a technique that separates molecules based on their electrical charge. By comparing hemoglobin from healthy individuals with hemoglobin from individuals with sickle cell anemia, they sought to identify subtle differences in their electrical properties. The key experiment involved isolating hemoglobin from both normal individuals and those afflicted with sickle cell anemia. These samples were then subjected to electrophoresis. The results revealed a distinct difference in the electrophoretic mobility of the two hemoglobin types. Normal hemoglobin migrated at a different rate than sickle cell hemoglobin, indicating a difference in their electrical charge. This difference in charge suggested that the amino acid composition or arrangement of the hemoglobin molecule was altered in individuals with sickle cell anemia. A specific example of this work is Pauling's publication in 1949, co-authored with Harvey Tarnow, S.J. Sainer, and Ibit Wells in the journal Science. The article, titled Sickle Cell Anemia, a Molecular Disease, presented compelling evidence that sickle cell anemia is caused by a difference in the electrical charge of hemoglobin. This publication marked a turning point in the understanding of genetic diseases, establishing the concept of a molecular disease where a specific molecular defect is directly responsible for a disease phenotype. The paper detailed the experimental evidence from electrophoresis, demonstrating the altered mobility of sickle cell hemoglobin compared to normal hemoglobin. The conclusion was unequivocal: sickle cell anemia is a disease at the molecular level. Archibald Garrett's earlier concept of inborn errors of metabolism exerted a direct influence on Pauling's thinking about the molecular basis of disease. Garrett, a British physician, proposed in the early 20th century that certain diseases, such as alpha-ptenuria, were caused by inherited defects in specific enzymes. These defects disrupted metabolic pathways, leading to the accumulation of abnormal metabolites. Garrett's work provided a conceptual framework for understanding how genetic mutations could lead to biochemical abnormalities and disease. Pauling extended Garrett's ideas by focusing on the structure of proteins, proposing that mutations could alter the shape and function of proteins, leading to a wide range of diseases. Pauling explicitly acknowledged Garrett's influence in his writings on sickle cell anemia. Max Perritz's later work on the detailed structure of hemoglobin provided further confirmation of Pauling's hypothesis about the molecular alteration in sickle cell anemia. Perritz, using X-ray crystallography, determined the three-dimensional structure of hemoglobin and revealed the precise location of the mutation responsible for sickle cell anemia. He found that a single amino acid substitution, the replacement of glutamic acid with valine at a specific position in the beta-globin chain, caused the hemoglobin molecules to aggregate and form long fibers, leading to the characteristic sickle shape of red blood cells. Perritz's structural data provided a detailed molecular explanation for the phenomenon Pauling had observed through electrophoresis. The work of Perritz served as a powerful validation of Pauling's initial hypothesis. The influence of Pauling's work on sickle cell anemia extended to numerous scientists. Vernon Ingram, for example, directly built upon Pauling's findings. Ingram used a technique called fingerprinting to identify the exact amino acid difference in sickle cell hemoglobin. His work pinpointed the specific amino acid substitution responsible for the disease. Ingram's research provided a crucial piece of the puzzle, confirming Pauling's hypothesis and paving the way for a deeper understanding of the molecular basis of sickle cell anemia. Modern relevance lies in Pauling's work on sickle cell anemia, laying the foundation for the field of molecular medicine and our understanding of genetic diseases at the molecular level. Prior to Pauling's work, the link between genes and disease was largely conceptual. He demonstrated that genes could directly influence the structure and function of proteins, and that alterations in protein structure could lead to disease. This concept revolutionized the understanding of disease and opened up new avenues for diagnosis and treatment. This work directly led to the development of genetic screening and gene therapies. Genetic screening allows for the identification of individuals who carry the sickle cell gene or are at risk of developing the disease. This information can be used to provide genetic counseling and to offer preventative measures. Gene therapies, which aim to correct a genetic defect responsible for sickle cell anemia, are currently under development. These therapies hold the promise of curing the disease by replacing the mutated gene with a normal copy. The development of these tools and therapies owes a great debt to Pauling's pioneering work on sickle cell anemia. His insights transformed our understanding of disease and paved the way for a new era of molecular medicine. Pauling's exploration of the molecular basis of disease, exemplified by his work on sickle cell anemia, laid the groundwork for advancements in genetic screening and gene therapies. His insights into the relationship between protein structure and function transformed the understanding of disease and spurred the development of new diagnostic and therapeutic approaches. Following his groundbreaking work on chemical bonding, molecular structure, and the nature of proteins, Pauling embarked on a path that would generate considerable controversy within the scientific community and beyond. His advocacy for high doses of vitamin C. He became convinced that large quantities of ascorbic acid or vitamin C could offer protection against a wide range of ailments, from the common cold to cancer. This conviction, while passionately held, met with significant skepticism from many of his peers. The problem Pauling addressed was the perceived inadequacy of the recommended daily allowance of vitamin C for maintaining optimal health. He posited that humans, unlike most other mammals, are unable to synthesize vitamin C internally and therefore are particularly susceptible to deficiency. He reasoned that significantly higher intakes than those typically recommended could enhance the immune system and provide protection against disease. Pauling's methodology in this area differed significantly from the rigorous, controlled experimental approaches that characterized his earlier work. His advocacy for vitamin C was based largely on anecdotal evidence, personal experience, and some clinical studies. These studies, however, often faced criticism for their lack of proper controls, small sample sizes, and potential biases. A specific example of Pauling's advocacy is his 1970 book, Vitamin C and the Common Cold. In this book, he argued that taking large doses of vitamin C could significantly reduce the incidence and severity of colds. The book became a bestseller, fueling public interest in vitamin C and leading to a surge in its consumption. However, the scientific community reacted with considerable skepticism. Numerous controlled clinical trials were conducted to test Pauling's claims, and the results were mixed. While some studies suggested a modest reduction in the duration or severity of cold symptoms with high doses of vitamin C, others found no significant benefit. Pauling's interest in vitamin C was sparked, in part, by the work of Albert Szent-Györgyi, who had discovered vitamin C and elucidated its role in fundamental metabolic processes. Szent-Györgyi's research highlighted the importance of vitamin C as an antioxidant and its involvement in various enzymatic reactions. Pauling built upon this foundation, proposing that even higher doses of vitamin C could provide additional benefits by further enhancing antioxidant defenses and supporting immune function. It is important to note that Szent-Györgyi himself later expressed reservations about Pauling's claims regarding the efficacy of high-dose vitamin C. The contrast between Pauling's approach and that of many other researchers in this area is striking. While Pauling relied heavily on anecdotal evidence and less rigorous studies, others employed more controlled clinical trials with larger sample sizes and blinding techniques to minimize bias. These studies often failed to confirm Pauling's claims of substantial benefits from high-dose vitamin C. One notable example is the work of Terence Anderson at the University of Toronto. Anderson conducted several well-designed clinical trials to assess the effect of vitamin C on cold symptoms. His findings suggested that vitamin C had, at best, a modest effect on the duration and severity of colds, a conclusion that contrasted sharply with Pauling's more optimistic claims. The reception of Pauling's vitamin C advocacy was mixed. While the public embraced his ideas, generating a surge in vitamin C consumption, the scientific community remained largely unconvinced. Many researchers criticized his methodology and questioned the validity of his claims. The controversy surrounding vitamin C continued for many years, with ongoing debates about its potential benefits and risks. It is worth noting that Pauling's advocacy for vitamin C stimulated further research into the role of micronutrients in health. While his specific claims about vitamin C may have been overstated, his work helped to raise awareness of the importance of nutrition in maintaining overall well-being. Subsequent research has explored the potential benefits of vitamin C and other micronutrients in various health conditions, leading to a more nuanced understanding of their role in human health. In reflecting on Linus Pauling's remarkable scientific career, one finds a tapestry woven with threads of brilliance, innovation, and, in his later years, controversy. His groundbreaking contributions to the understanding of chemical bonding, molecular structure, and the nature of proteins cemented his place as one of the most influential scientists of the 20th century. His application of quantum mechanics to chemistry revolutionized the field, providing a theoretical framework for understanding the behavior of molecules. His elucidation of the alpha helix and beta sheet structures of proteins transformed the field of molecular biology, providing insights into the relationship between protein structure and function. His work on sickle cell anemia demonstrated the molecular basis of disease, paving the way for advancements in genetic screening and gene therapies. Pauling's legacy stands as a testament to the power of scientific curiosity, intellectual rigor, and the pursuit of knowledge. His pioneering work continues to inspire scientists and researchers around the world. He shaped our understanding of the fundamental principles governing the behavior of matter, and he left an indelible mark on the fields of chemistry, molecular biology, and medicine. While his later advocacy for high doses of vitamin C generated considerable controversy, it also stimulated further research into the role of micronutrients in health, contributing to a more comprehensive understanding of the relationship between nutrition and well-being. His life serves as a reminder that even the most brilliant minds can be subject to error, but that the pursuit of truth, however challenging, remains the ultimate goal of scientific endeavor. Pauling's impact on science is undeniable, and his work will continue to be studied and debated for generations to come.
We really appreciate you tuning in to this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.